Hello, everyone, and welcome to today's poster presentation, Development of Multiplex xMAP Technology-Based Assays for Simultaneous Detection of Soluble Checkpoint Molecules Involved in Anti-Cancer Immune Response, presented by Suzanne Oehler, Manager, Research and Development, Antibodies and Immunoassays, Thermo Fisher Scientific. I'm Alexis Krause of LabRoots, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by Thermo Fisher Scientific. For more information about our sponsor, please visit www.thermofisher.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. We'll answer as many of your questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please use the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Suzanne Oehler. I will now turn the presentation over to her.
Thank you, Alexis. In today's talk, I will be presenting a poster about development of multiplex xMAP technology bead-based assays for the simultaneous detection of soluble checkpoint molecules involved in anti-cancer immune response. At first, I would like to give you some background information about tumor immunology. Our immune system has evolved to protect the integrity of multicellular organisms against external threats, such as pathologic organisms on the one hand, but also against malignant cells that are generated in cancerogenesis. During cancerogenesis, genetic and epigenetic alterations of the cells occur, which the immune system can use to distinguish tumor cells from their normal counterparts. T cells and NK cells are important players in fighting malignant cells. Harnessing the power of the immune system to find cancer is a therapeutic approach which is very successful for certain cancer types and individual patients. Many new anti-cancer therapies aiming to engage the power of the immune system to combat cancer are under investigation. However, tumor cells also acquire escape mechanisms to avoid being attacked by the immune system, which may lead to immune resistance. Under physiological conditions, the activity of immune cells needs to be strictly controlled to ensure the immune system's balance between the recognition of non-self and the prevention of autoimmunity. This regulation involves proteins, referred to as immune checkpoints, which can affect immunoregulatory pathways by either boosting or restricting the immune response. Here you see the cancer immunity cycle as proposed by Chen and Melman. This cycle describes how the immune system reacts to malignant cells and the interactions that take place between immune cells and tumor cells. I will walk you through this cycle. Let us begin with number 1, which is below. So, tumor cell antigens, so-called neoantigens, are released to the microenvironment. The neoantigens are phagocytosed by antigen-presenting cells like dendritic cells, which present the neoantigen and migrate to the lymph node. In the lymph node, the neoantigens are presented to T cells. The T cells get primed and activated and start to proliferate. The activated T cells migrate via blood vessels to the tumor and infiltrate the tumor. This is called extravasation. The activated T cells recognize their target in the tumor via the T cell receptor. This is shown in number 6. And finally, the cancer cell gets killed by cytotoxic T cells. This is in step number 7. And the killing of the cancer cells lead to further release of more neoantigens. Now we are at number 1 again. Thus, this leads to a propagation of the immune response. Each step of this cycle requires the coordination of numerous factors, both stimulatory and inhibitory in nature. Stimulatory factors are shown in green here, promote immunity, whereas inhibitors, shown in red, reduce immune activity to avoid overactivation of the immune system, causing damage to healthy cells. These regulating factors may be cytokines, factors released by dying tumor cells and the microbiota, and checkpoint molecules. It is important to note that not only tumor cells but also other cell types of the immune system like intratumoral T regulatory cells, macrophages, and myeloid-derived suppressor cells are sources of inhibitory factors. If the inhibitory factors outweigh the stimulatory factors, the tumor can escape the host's immune response. The goal of immunotherapy is to initiate or reinitiate a self-sustaining cycle of cancer immunity, enabling it to amplify and propagate, but not so much as to generate unrestrained autoimmune inflammatory responses. Immune checkpoint molecules are recognized as intact transmembrane proteins. However, soluble isoforms or shed variants have been identified for the majority of these molecules. These soluble checkpoint molecules are either generated by shedding from the membrane via proteases, predominantly ADAM proteases, or they are generated as soluble variants by alternative splicing. Soluble checkpoint molecules may function as immune adjuvants or decoy receptors, and they may thereby regulate the checkpoint pathways. To provide a tool for the measurement of circulating soluble checkpoint molecules, we developed a Procataplex panel, which is based upon Luminex beads technology for the targets. The targets you see listed here in this slide. High plasma concentration of these soluble molecules may interfere with the efficiency of cancer therapies using antibodies against checkpoint modulators of T cells. In addition to the important role of the adaptive immunity, also innate immune cells, especially NK cells, contribute to anti-cancer immune response. The major role of NK cells is to recognize virus-infected cells, which show alterations on their cell surface. In analogy, NK cells can also recognize and attack transformed malignant cells lacking MHC class I. In addition to MHC class I deficiency, NK cell activity is mediated by multiple activating and inhibitory receptors, which determine if NK cell becomes cytotoxic. Here you see the receptors and ligands either on the tumor cell or on the NK cell. Activated NK cells can act directly via perforin, which disrupts the target cell membrane. In addition, NK cells may also act indirectly by secreting pro-inflammatory cytokines, thereby stimulating adaptive immune response such as T cell response. activating receptors of NK cells described so far are NKG2D, NCR, and DNAM1, which can act in a synergistic way. Ligands to NKG2D, like MIC-A, MIC-B, and UL16 binding proteins, are upregulated in cellular stress response and expressed by many tumor cells. However, during tumor progression, cancer cells develop immunosuppressive mechanisms that circumvent NK cell-mediated killing. Shedding soluble ligands for NK cell activating receptors is a mechanism tumor cells use to escape immunosurveillance. Therefore, similarly as for T cells, immunotherapeutic agents are under investigation for NK cells to stimulate their lytic capacity and support them to recognize and eradicate tumors. The major NK cell receptors that are potential immunotherapeutic targets are shown in this slide here. And here on the next slide, you have receptors and ligands from NK cells, which can be measured with our immuno-oncology checkpoint panel 2. On this slide, the targets are listed. For example, the soluble forms of the already mentioned MIC-A and MIC-B. Both immuno-oncology checkpoint panels can be combined and also extended with cytokines, chemokines, and adhesion molecules, which encounter another important role in shaping local and systemic immune responses. Various cytokines have been demonstrated to be associated with chronic inflammation, which may promote cancer development, or with acute inflammation, which facilitates cancer rejection. The simultaneous detection of immune stimulatory and inhibitory factors allows to gain a more holistic picture of the molecular players in cancer immunity and should help to shed light on the biology of the pathways, as show first results of our customers using these panels. In the next slides, I will show you some examples from our customers. This data you see here was generated by one of our beta testers using both immuno-oncology checkpoint panels in combination. As samples, sera from melanoma patients were analyzed. For all targets of both panels, measured levels are shown here. The levels are shown as mean values of about 70 to 90 samples. As you can see, there were detectable levels for soluble checkpoint marker proteins in these samples. Here, the next example shows the effect of treatment on one of these targets. The samples are grouped in pre-treatment, which you see on the left, and early on treatment on the right, and the mean PD-1 level is shown for each group. During treatment, the mean serum concentration of PD-1 decreases strongly, as you can see. Here you see the levels of individual patients. For every patient, the PD-1 level before treatment, this is shown in blue, and during treatment, shown in red, is shown here. So, as you can see, the levels of the individual samples differed strongly. These data are promising but preliminary, and it will be very interesting to combine the data obtained with clinical outcome of therapy. Here you see data obtained with our immuno-oncology panel during beta testing, which is already published by our beta tester. the detected levels of several checkpoint markers in samples from human breast cancer patients before and after chemotherapy are shown. 6 immune checkpoint molecules were detected with high level in these samples. Interestingly, CD27, CD80, and CD273 correlated with the HMGB1 level and were increased in human samples with a better response to epirubicin docetaxel chemotherapy and a better 5-year survival. Time-dependent changes of specific soluble checkpoint molecules can be seen here in our last example. This data is obtained by measurement we did in-house with samples which were provided by our PETA tester. In this study, the immuno-oncology panel in combination with the TNF receptor superfamily panel was used to measure samples taken at different time points during therapy. As an example, the data for CD27 is shown on the left, and on the right side you have the data for CD30. While CD27 levels were very consistent over time, for CD30 there was an increase at the beginning of the treatment, which went down again during treatment. In addition to our Multiplex immuno-oncology checkpoint marker panels, we also have developed ELISAs for immuno-oncology targets. These available ELISAs are listed on this slide. Our beta testing results confirm that our Multiplex immuno-oncology checkpoint marker panels can be used to detect different expression patterns of soluble markers dependent on disease state and cancer treatment. The systematic simultaneous analysis of these soluble biomarkers should help to shed light on the biology of the pathways and possibly help to better understand the response to therapeutic treatment. Thank you very much for your attention. I will take any questions now.
Thank you, Dr. Oehler, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on the screen and click the Send button. We'll answer as many of your questions as we have time for. So let's get started. Our first question is, what is the sample volume needed for analysis? Are frozen samples useful? And how many samples can be analyzed with one kit?
The sample volume which is needed is 25 microliter. This is enough for measuring all analytes in combinable panels. So it's really not a lot of amount which is needed. Frozen samples can be used, and with one kit there can be measured up to 80 samples. Also, since we recommend to do testings in parallel for each sample, it would be then reduced to 40 samples per kit.
Now our next question is, what is the sensitivity of your Multiplex assays?
The sensitivity, of course, is depending on the individual analyte. For our immuno-oncology checkpoint marker, we have a low limit of quantification in the lower picogram per ml range.
And it looks like we have time for one more question. So what can I do if I need to measure checkpoint markers in mouse models? Is your panel cross-reacting with a mouse?
No, as far as we know, it doesn't cross-react. We did testings with a few purchased mouse serum pools, and these did not show any levels. So we can assume it's not cross-reacting. But at present, we have developed, or we are at the end of developing, mouse immuno-oncology targets, which will be combined in 2 panels. We will have a 4-plex, which contains LAG3, TIM3, BTLA, and CD27. And then there will be another panel, a 7-plex, which contains CD80, CTLE4, PD1, PDL1, PDL2, CD137L, and DCH3. These both panels will not be combinable, but they can be measured. They can be used as panels to measure these analytes. This is what we are going to, what we can provide for mouse analytes.
I would like to once again thank Dr. Oehler for her presentation. I would also like to thank Thermo Fisher Scientific for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through September of 2019. Please share that with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.